Metabolic engineering is the practice of optimizing genetic and regulatory processes within cells to increase the cell's production of a certain substance. These processes are chemical networks that use a series of biochemical reactions and enzymes that allow cells to convert raw materials into molecules necessary for the cell's survival. Metabolic engineering specifically seeks to mathematically model these networks, calculate a yield of useful products, and pinpoint parts of the network that constrain the production of these products. Genetic engineering techniques can then be used to modify the network in order to relieve these constraints. Once again, this modified network can be modeled to calculate the new product yield. The ultimate goal of metabolic engineering is to be able to use these organisms to produce valuable substances on an industrial scale in a cost-effective manner. Current examples include producing beer, wine, cheese, pharmaceuticals, and other biotechnology products. Some of the common strategies used for metabolic engineering are 1. Overexpressing the gene encoding the rate-limiting enzyme of the synthetic pathway, 2. Blocking the competing metabolic pathways, 3. Heterologous gene expression, and 4. Enzyme engineering. Since cells use these metabolic networks for their survival, changes can have drastic effects on the cell's viability. Therefore, trade-offs in metabolic engineering arise between the cell's ability to produce the desired substance and its natural survival needs. Therefore, instead of directly deleting and/or overexpressing the genes that encode for metabolic enzymes, the current focus is to target the regulatory networks in a cell to efficiently engineer the metabolism. History and applications. In the past, to increase the productivity of a desired metabolite, a microorganism was genetically modified by chemically induced mutation, and the mutant strain that overexpressed the desired metabolite was then chosen. However, one of the main problems with this technique was that the metabolic pathway for the production of that metabolite was not analyzed, and as a result, the constraints to production and relevant pathway enzymes to be modified were unknown. In 1990s, a new technique called metabolic engineering emerged. This technique analyzes the metabolic pathway of a microorganism and determines the constraints and their effects on the production of desired compounds. It then uses genetic engineering to relieve these constraints. Some examples of successful metabolic engineering are the following: identification of constraints to lysine production in Corynebacterium glutamic, insertion of new genes to relieve these constraints to improve production, e.e. engineering of a new fatty acid biosynthesis pathway, called reversed beta oxidation pathway, that is more efficient than the native pathway in producing fatty acids and alcohols which can potentially be catalytically converted to chemicals and fuels, improved production of DAHP and aromatic metabolite produced by E. coli that is an intermediate in the production of aromatic amino acids. It was determined through metabolic flux analysis that the theoretical maximal yield of DAHP per glucose molecule utilized was 3/7. This is because some of the carbon from glucose is lost as carbon dioxide, instead of being utilized to produce DAHP. Also, one of the metabolites PEP, or phosphoenolpyruvate, that are used to produce DAHP, was being converted to pyruvate PYR to transport glucose into the cell, and therefore, was no longer available to produce DAHP. In order to relieve the shortage of PEP and increase yield, Padnaik et al. Used genetic engineering on E. coli to introduce a reaction that converts PYR back to PEP. Thus, the PEP used to transport glucose into the cell is regenerated, and can be used to make DAHP. This resulted in a new theoretical maximal yield of 6/7, double that of the native E. coli system. At the industrial scale, metabolic engineering is becoming more convenient and cost-effective. According to the Biotechnology Industry Organization, more than 50 biorefinery facilities are being built across North America to apply metabolic engineering to produce biofuels and chemicals from renewable biomass which can help reduce greenhouse gas emissions. Potential biofuels include short-chain alcohols and alkanes to replace gasoline, fatty acid methyl esters and fatty alcohols to replace diesel, and fatty acid and isoprenoid-based biofuels to replace diesel. Metabolic engineering continues to evolve in efficiency and processes aided by breakthroughs in the field of synthetic biology and progress in understanding metabolite damage and its repair or preemption. Early metabolic engineering experiments showed that accumulation of reactive intermediates can limit flux in engineered pathways and be deleterious to host cells if matching damage control systems are missing or inadequate. Researchers in synthetic biology optimize genetic pathways, which in turn influence cellular metabolic outputs. Recent decreases in cost of synthesized DNA and developments in genetic circuits help to influence the ability of metabolic engineering to produce desired outputs. Metabolic flux analysis. Setting up a metabolic pathway for analysis. The first step in the process is to identify a desired goal to achieve through the improvement or modification of an organism's metabolism. Reference books and online databases are used to research reactions and metabolic pathways that are able to produce this product or result. These databases contain copious genomic and chemical information including pathways for metabolism and other cellular processes. Using this research, an organism is chosen that will be used to create the desired product or result. Considerations that are taken into account when making this decision are how close the organism's metabolic pathway is to the desired pathway, the maintenance costs associated with the organism, and how easy it is to modify the pathway of the organism. Escherichia coli (E. coli) is widely used in metabolic engineering to synthesize a wide variety of products such as amino acids because it is relatively easy to maintain and modify. If the organism does not contain the complete pathway for the desired product or result, then genes that produce the missing enzymes must be incorporated into the organism. Determining the optimal genetic manipulation.